All right, so now we need to talk about muscle physiology. Here you won't find the corresponding PowerPoint presentation on your Blackboard page because I don't find that it's particularly conducive to this. It helps if you walk through it step by step and you sort of take notes as we go along. So to begin with, muscle is one of our four primary tissue classes. We've got epithelial tissue, we've got connective tissue, and then we have muscle and neurons. So muscle is a tissue class all by itself. Now there are three different types of muscle. We have skeletal muscle, cardiac muscle, and smooth muscle. Now, cardiac muscle and smooth muscle we will talk about in their own rights, and there should be a corresponding PowerPoint actually for those two muscle types. We're going to touch on that and discuss the differences between cardiac muscle and smooth muscle and skeletal muscle later. But for the moment, we're going to focus primarily on skeletal muscle. So, skeletal muscle. Some qualities of skeletal muscle. First of all, skeletal muscle is the only one of those three muscle types that is voluntary. Voluntary meaning that you have conscious control over it. You can make it contract. Conscious thought. You don't get to do that with your heart or with smooth muscle. Skeletal muscle is voluntary. Skeletal muscle is striated. What that means is that you'll see light and dark banding. If we look at skeletal muscle on a microscope slide, what you'll see is striations. So it is striated. Skeletal muscle. All right, so voluntary muscle, striated muscle. Let's talk about qualities of muscle as a whole, and this actually applies to all muscle types, not just skeletal. Those are qualities of just skeletal muscle, but all muscle does have some qualifications. First of all, muscle is contractile. Muscle works by getting shorter. The cell decreases in length, and that's how the muscle does work. That's contractile. Muscle tissue is conductive, which means that it will conduct an electrical impulse. And we'll talk about that electrical impulse in just a bit. Muscle tissue is extensible, which means you can stretch it out. It will get longer. That's not how it works. Muscle tissue doesn't push, it pulls, but you can extend those fibers, you can stretch them out. And when you do that, they will rebound back to their original shape. It is also elastic. And our last characteristic here is that it is excitable. Excitable means that it responds to a stimulus, in this case, that electrical impulse that we'll talk about in just a bit. That's the stimulus that makes it contract. So all muscle tissue has these types. Now there are some terms that we'll use here as we discuss muscle that these terms don't apply to other tissues. First of all, the muscle cell is called a fiber. Now, skeletal muscle fibers are very long cells. Skeletal muscle fibers, and again, so we're just referring to skeletal muscle here, are multinucleate. Meaning they have more than one nucleus. You get my point. So more than one nucleus. That also means that skeletal muscle fibers are, amitotic. Skeletal muscle fibers don't undergo mitosis. Mature skeletal muscle fibers don't undergo mitosis. It heals through fibrosis, not regeneration. other terms here. The plasma membrane, or the cell membrane, is called the sarcolemma. Anytime you see that suffix sarco always refers to muscle. So sarcolemma, it's the cell membrane. The smooth endoplasmic reticulum is called the sarcoplasmic reticulum. And the function for the smooth ER here is a bit different than we see in other cells. We'll discuss the sarcoplasmic reticulum and that function in just a second. You get the point here. So, right, anytime we see muscle, we'll either have sarco as a suffix. The other suffix that indicates muscle is myo. Myo always means muscle as well. So, as we discussed in the very beginning, one of the big themes of anatomy and physiology is the hierarchy of structure. And really, muscle is one of the better examples of this principle of hierarchy of structure. So, your muscle is capable of tremendous amounts of force. Your muscles can actually generate more force than your bones can handle. Now, we don't do that. Even under maximum effort, typically you don't use more than about a third of the fibers in any muscle simultaneously, because you don't want to damage the skeletal system in this movement. That being said, muscles are still made up of cells, which are microscopic, and the movement that's going to occur that we'll look at in just a little bit is right on the subcellular level. We're looking at tiny little elements of the cytoskeleton slightly sliding past one another. How then do we produce so much force? Well, we do that with this hierarchy of structure. We start with very, very small elements and multiply those and bundle them together and multiply that and bundle them together, so that even though there's these very tiny elements moving slightly past one another, when you multiply that by millions, if not hundreds of millions of times, you can produce a lot of force. So the smallest element here within the muscle fiber is called a myofilament. And there are actually two myofilaments. We have a thick filament and we have a thin filament. All right, so myofilaments, the smallest bundle. Myofilaments are bundled into myofibrils. So a myofibril is a bundle of myofilaments. Then myofibrils are bundled together into the muscle fiber. The muscle fiber, then, muscle fibers are bundled into things called fascicles. And then the fascicle is bundled into the whole muscle. So a muscle is a bundle of fascicles. A fascicle is a bundle of fibers. If you've ever eaten any sort of meat, like the little strings of meat that you get, those are fascicles. If you eat like a steak, you can see the different muscle groups in that steak. They're a little bit more defined, and then the little individual small pieces there are fascicles. You can't really see the cells because they're microscopic. In addition to these elements, there are connective tissue wrappings that separate them. Each muscle fiber, for instance, is surrounded by a connective tissue called the endomysium. The fascicles are surrounded by another tissue called the perimysium. And the muscle itself is surrounded by yet another connective tissue called the epimysium. So the epimysium separates muscles from one another. The perimysium separates fascicles from one another. And the endomysium separates fibers from one another. Remember that muscle tissue is excitable tissue. These are connective tissues. Muscle tissue is also contractile tissue. These are connective tissues. They are neither excitable nor contractile. In fact, collectively, these three things are referred to as the evidently I just discovered another function on this pen. Contractile elements. They are these three connective tissue wrappings. And they separate those elements from one another so that we do have this fine degree of motor control. We can contract muscle without contracting the whole muscle because the elements are separated from one another by these connective tissues. Now, these connective tissues are elastic slightly, so they do provide some of the rebound that moves the muscle back to its original position. So here's our hierarchy of structure. So now, before we move any further, well, before I do that, let's talk about this idea of myofilaments and myofibrils and that sort of thing. Our unit striated muscle. Oh, crap. It's called a sarcomere. All right, so the sarcomere. So let's do this. Oops. Sorry, having issues. Not sure what I've done here, but it's not drawing on me anymore. All right, so hopefully I've got this worked out. Let's do the sarcomere. Oops. Undo. Don't know what I did there either. All right, so let's label some things here. So here, called the Z disc. It's like a zigzag, right? On this thing, this is the M line. Anchor there in the middle. Now we've got this gap that's right here. That gap is called the H zone. Here, we've got that big fat blue line. That is your A band. And then here, in between those big fat blue lines, that is your I band. Now, these blue ones, that would be your thick filament or your thick myofilament. And that means these red ones then would be your thin myofilaments. Now, it's the movement that takes place here at the sarcomere that's going to be the movement that causes muscle contraction. When muscles contract, the Z disc will move closer together because the proteins of that thick filament are going to latch onto the proteins of that thin filament, and they're going to pull them in toward that M line in the middle. So when muscle contracts, the Z disc get shorter together. The I band and A band here, this is what makes muscle striated. The I band, and I wish I was kidding here, the I band stands for light. Every stupid book you'll see will have that. And the A band, of course, then stands for dark. I don't know who came up with that. And that's what we see. When the muscles relax, you have a light band and a dark band. Anywhere there's a thick filament, a dark band. Anywhere there's a thin filament, you've got that light band. So this is the sarcomere. Just as a heads up, you'll need to be able to label these parts of a sarcomere for your exam. And later when we look at lab stuff, I've actually got some sarcomere models here that will look at the movement and what's going on at the sarcomere. But like I said, it's the interaction between these thin and thick filaments that's going to create that contraction of muscle. Now, like we said before, muscle tissue is excitable. And it's conductive. This all involves this idea of this electrical impulse, and that's actually what we need to talk about before we look at what's going on with the motion of the muscle. This tends to be why muscle is more complicated, because we don't just have this physical motion, we have this electrical issue going on as well. So think back to when we talked about cells and when we talked about potential, the difference in charge between the inside and the outside of the cell. Remember, you've got that sodium-potassium pump. We're pumping 3 sodium out and 2 potassium back in, so that we get potential. We get a difference in charge between the inside and the outside of the cell. We're going to graph that here at like negative 70 millivolts. That negative 70 millivolts is your resting membrane potential. The charge on that cell membrane at rest. going to put another number here. Negative 55 millivolts. That is going to be, hypothetically, that is going to be, it's a terrible place for that. I'm going to redraw my graph, so bear with me. So negative 70, our resting membrane potential. Negative 55 is going to be our threshold. Way up here at, let's say, 35, positive 35 millivolts. We have sort of the max here. So what we've got to do is we've got to create some sort of current, some sort of movement of this potential. We've got to use that difference in charge, and we're going to do that by opening sodium and potassium channels. We put a bunch of sodium out here, we put a bunch of potassium in here. That means sodium wants to come in and potassium wants to go out. If I let sodium in, what happens to the charge? It goes up. If I let potassium out, what happens to the charge? It goes down. This happens very, very quickly. And this rapid depolarization. Depolarization means we go from that positive to negative. So this upward, excuse me, this upward going from negative to positive, that's depolarization. And then repolarization, going back the other way, down. So going back down, that's repolarization. Of that membrane is called the action potential. And for our purposes, this action potential is kind of like electrical current. Now, I don't want you to confuse this with electricity because it's not the same thing as just straight up electricity. We'll talk more about action potential when we get to the nervous system. But right now, this is sort of the electrical energy that's going to make the muscle contract. And that electrical energy is going to open and close different channels that make other stuff happen. So keep this in mind. It's easier now if we just go ahead and talk about the neuromuscular junction and then go back and touch this as necessary. I just needed you to kind of get a slight understanding of action potential. We're going to talk about action potential in depth and crazy detail when we get to the nervous system. Right now, it's current. Just keep that. So now let's look at the neuromuscular junction. As the name suggests, the neuromuscular junction is the junction between the neurons, the nervous system, and the muscle. Oops, I didn't mean to do that. Right. All right. Now, in skeletal muscle, every skeletal muscle has a neuromuscular junction. So let's draw this. Here we have our neuron. This part of the neuron is called the axon. And this part is called the synaptic bulb. Down here we'll make our muscle fiber. So this would be the sarcolemma, the cell membrane, the muscle cell. This little part of the sarcolemma where you have this little indentation is called the motor end plate. Now on that motor end plate, we're going to have these little things. These are ligand gated ion channels. More on that in a bit. Eraser tool. This thing is called the T-tubule. That T is for transverse because it kind of cuts across the muscle cell. And on either side of that T-tubule, I'm going to have this structure, expertly drawn, I might add, happy trees. And this will be, oh, that's grotesque. Hold on. I have enough OCD about me to care, I guess. That's not much better. We're going to scope it. All right, so this then is your Sarcoplasmic Reticulum. Normally in our in-person class, it's at this point that I actually inform everyone in class that for the last 40 years, my dad has taught drafting. And I don't know how I passed that class. Probably just out of pity. Because as you can tell, not my strong suit. Anyway, so we've got our major parts here. Now, what we're going to do is we're going to walk through our events that are going to occur here at the neuromuscular junction on a step-by-step thing. So let's start at the beginning. First, the first thing that happens is an action potential arrives at the synaptic bulb. Now this is voluntary, right? This is from the nervous system, the brain, the spinal cord. So that electrical impulse travels down that axon and then gets the synaptic bulb. When it gets to the synaptic bulb, what's going to happen is that it's going to open voltage gated calcium channels. Just to make sure that's clear. That's not any better. Hold on. This is so much better. Calcium channels. Voltage gated calcium channels. And then calcium enters the synaptic bulb and acetylcholine goes out. So that electrical impulse goes all the way down here. It opens voltage-gated calcium channels. So a voltage-gated channel means that voltage is necessary to open the channel. It's like an electric door, right? It requires electricity to open. Here, those electric doors open, and it's going to let calcium in. So calcium that was on the outside goes in. That's a T positive. Calcium goes in. And when calcium goes in, a neurotransmitter goes out. Our neurotransmitter here is acetylcholine, or ACH. So acetylcholine goes out. Now, acetylcholine binds to receptors. These ligand-gated ion channels are acetylcholine receptors. A ligand is anything that binds to a receptor. So in this case, our ligand is acetylcholine. They're acetylcholine-gated ion channels. And when acetylcholine binds to its receptor, when that neurotransmitter comes across and binds to its receptor, it opens those channels and we get sodium influx. So those channels open, there's acetylcholine binding to its channel. And that's going to let sodium come in. Now, if we're going back and we're looking at our potential, we'll scroll back up here real quick, what I erased. Sodium coming in, that's depolarization. So you start to get this movement where we go from that negative 70 millivolts, starting going up toward negative 55. When there's enough sodium in the cell that it hits that negative 55, when we reach the threshold potential, so threshold is reached, that triggers an action potential on the muscle fiber now. So the threshold is reached, that triggers action potential on the muscle fiber. And now that action potential is going to travel along the sarcolemma, the cell membrane, and it's going to go down that T-tubule and back up and out. So action potential travels across the membrane down the T-tubule. Now when it goes down that T-tubule is touching that sarcoplasmic reticulum. And they're always found in groups of 3 like that, 1, 2, 3. And as the action potential goes down that T-tubule, it opens voltage-gated calcium channels on the sarcoplasmic reticulum. Now, in the muscle fiber, this smoothie or the sarcoplasmic reticulum, its function is to store calcium. There's calcium in here. When those channels open, that calcium leaves. Of this, we get an increase in intracellular calcium. That is why all this crap happened. We were through all these steps, all this opening of channels and neurotransmitters and action potentials and all this crap just to get that, to get the increase in intracellular calcium. That increase in intracellular calcium is your final trigger for contraction. And actually, this is true for all muscle types, not just skeletal muscle. More on that later. But this is your trigger for contraction. So why? Why is that your trigger for contraction? What could possibly happen? What does that calcium do? Remember what I said was the actual mechanism here, the filaments sliding past one another. The calcium triggers that. So now let's look at those filaments. Let's zoom in on that sarcomere, those thin and thick filaments. So here is a close-up view of our thin myofilament. A thin myofilament is made of a protein called actin. So you'll see it referred to as the actin myofilament. Actin's always very sad. The little sad face places on actin are called binding sites. A binding site is obviously where it can bind to something. Those binding sites are blocked by a string of protein called tropomyosin. So you should know that tropomyosin blocks the binding sites on actin. And then studied along that tropomyosin thing are these little beads of something called troponin. And that's our thin filament. Our thick filament is so many times bigger than this. We're not going to draw the whole thing. In fact, we're just going to draw a little part of it. Looks like this. Now, there are a bunch of these, and they have like these little tails, and they all come back together, and they're all combined. Again, not drawing. The thick filament is myosin. And these things are called the myosin heads. And stuck in each one of these myosin heads is a molecule of ADP and an inorganic phosphate. So ADP, inorganic phosphate. You get the point. So now, again, in a step-by-step manner, let's walk through what happens. What we have so far is we have this increase in intracellular calcium. We let all this calcium out. It's just floating around now. Calcium everywhere floating around. That calcium is going to bind to troponin. Calcium binds to troponin. And as we discussed way back in the beginning when we talked about proteins, when proteins bind to things, they change shape. When troponin and calcium stick together, troponin change shape. And that shape change, troponin, moves tropomyosin away from the binding sites. Oh man. On actin. So now those binding sites are exposed. Let's see. Make that work. Nope, not at all. Regardless, the binding sites are exposed. You get the idea. When those binding sites are exposed, myosin, don't get my Amazon stuff. Not at all. Swear to you. Binds to actin. So now that thin filament and that thick filament are stuck together. This myosin binds to actin. Seriously. It's called your cross bridge. Now, when proteins bind to stuff, they change shape. Myosin's a protein. When it binds to actin, that molecule of ADP, seriously, And that inorganic phosphate leave the myosin head. Because it sticks to actin. So they move out. So those guys take off, which causes the movement. Myosin head moves and it pulls actin with it. Because they're connected together, because they're formed, this cross bridge is formed, they're stuck together. And so when that myosin head moves, it pulls that actin in with it, and that moves those Z discs closer together, and now there's muscle contraction. The filament gets shorter because those cytoskeletal elements are pulling on each other. It's like grabbing a rope and pulling it in toward yourself. Those myosin heads grab onto it, and they start walking it in and pulling it in. When the myosin head moves, there's a vacancy that's been created by the exiting of that ADP and that inorganic phosphate. And an ATP molecule moves into the myosin head and that breaks the cross bridge. And now they separate. Then that ATP is broken down by an enzyme called an ATPase into an ADP, and that inorganic phosphate, and the cycle starts over. This is our cross bridge cycle. Myosin binds to actin. ADP and the inorganic phosphate get kicked out. The myosin head moves, it pulls the actin with it. ATP moves in, breaks the cross bridge, cycle starts over. As long as calcium is bound to troponin and that tropomyosin is not in the active site, the cycle begins again. And the muscle continues to contract. It's not synchronous. Those myosin filaments don't latch on in sync. They don't all grab on and then all let go. They take turns, and it's like they're walking along that filament in this step-by-step manner, and they pull it in toward that M line. They pull those Z discs closer together. Now, how do we relax the muscle then? Well, if we want to relax the muscle, we have to stop this. First thing, calcium can't be bound to troponin, because as long as calcium is bound to troponin, this cycle continues. So how do we stop that? Well, we go back here. And we have to get acetylcholine out of the synapse. How do we do that? Well, as luck would have it, we have an enzyme, acetylcholinesterase. Acetylcholinesterase breaks down acetylcholine. So then that acetylcholine is removed from the synapse. Then there's no more sodium going in. That means there's no more action potential here. That means there's no more calcium release here, which means that calcium is no longer bound to troponin. Troponin then isn't moving tropomyosin out of the way. The myosin's not binding to actin. Muscle relaxes. So there's a lot going on here. It's incredibly complicated. So much complexity. Why do we care? Because it is essential to being alive at any given moment. When you think about muscle, it's easy to think about, especially skeletal muscle, it's easy to think about it as voluntary movement, which it is. But one of those voluntary muscles is the diaphragm. And the diaphragm is the muscle that makes you breathe. Now, you don't have to think about it happening, but it is a voluntary muscle. You need proof, hold your breath, voluntary. So the diaphragm is a skeletal muscle, which means that this action is happening all the time, which means that if you screw this up, you stop breathing, which is bad for living. Fun fact, when you stop breathing, you stop living after a while, usually. Let's see what happens. So, what if, to begin with, what if I block that acetylcholine receptor? Incidentally, this would be called an antagonist. An antagonist is a blocker. So remember that the protein's three-dimensional shapes, right? We got that tertiary structure. And this is sort of the lock and key mechanism, right? Acetylcholine fits into that slot that's conveniently acetylcholine shaped, turns the lock, opens the gate. What if I don't know, pour superglue in that freaking lock? You're not putting a key in there anymore. And we can do that's an antagonist. It blocks it. A great example of an acetylcholine antagonist is called curare. Curare is a competitive antagonist of acetylcholine. Competitive means that it doesn't just block the acetylcholine receptor. It's like faster, it like races there and shoves acetylcholine into the ground and laughs at it and then binds to it. It's better binding to that site than acetylcholine is. But it doesn't turn the key. It doesn't unlock the lock. It just fits in the hole. So now it's blocked. If you block the acetylcholine receptor, what happens to the muscle? Nothing. Can't contract muscle if that receptor is blocked. Anything that prevents acetylcholine from binding prevents contraction. So add a little curare, what you get is paralysis. Now add a lot, you'll make whatever it is that got your curare dose, you'll make them stop breathing. That's good stuff. It's a reversible antagonist. It'll go away after a while. The understanding of how to block these acetylcholine receptors was a major advance in surgery. Before we understood how to do this, the surgeons had to be fast. One of the more infamous, I guess, surgeons bragged that he could amputate a limb in just a few seconds, and he could. And you had to, because I mean, you start cutting off some dude's arm, it doesn't matter how much you knock him out or how drunk he is, cutting off your arm sobers you up in a hurry. So he had to be fast. This guy was so fast that multiple times he amputated surgical assistants' fingers as well, because they didn't move. Yeah, great guy. But after we discovered how to do this, we can stop you from moving. Take your time. I'm not saying that we maintain unconsciousness or anything like that. That's a different sort of breed of anesthetic, but this would create paralysis. For the muscle to relax, acetylcholinesterase has to be in. So at the same time, anything that prevents acetylcholinesterase, hereby abbreviated as ACHE, from working, prevents relaxation. How important is that? That is so ridiculously important. Because if you stop acetylcholinesterase, it's not that you just prevent the muscle from relaxing, you make all the muscles contract. And remember I said that even under maximum effort, you don't contract all the muscle fibers. Now we override that. They all start contracting, including the diaphragm. How important is it? It's so important that we invaded another country, overthrew their government, and still occupy Iraq because we were looking for weapons of mass destruction that did just that. The weapons of mass destruction that we were looking for, things like VX gas and sarin gas, are acetylcholinesterase inhibitors. They're actually a group of chemicals called organophosphates. And while those two are weapons of mass destruction, we actually use organophosphates locally as pesticides. Things like malthion. Malthion's an organophosphate we spray on cotton, kills the hell out of bugs. But eventually, you'll have a patient that comes in because they were standing underneath the airplane when it flew over and they sprayed them. And they'll come in with muscle spasms because it's blocking acetylcholinesterase, difficulty breathing, muscle spasms, and you have to treat that. So, acetylcholinesterase causes the muscle to relax. Acetylcholine causes the muscle to contract. We'll look at neurotoxins later. We'll look at stuff like black widow venom. Black widow venom is an acetylcholine agonist. It acts like acetylcholine. And if you've ever been bitten, it hurts, but your muscle will spasm around the bite site because it's acting like acetylcholine, causing it to contract. So my question for you right now is, what happens when you die? I don't mean existentially, where your spirit goes with. I mean, like, if we were in person in class and you went out and you took a break and you didn't come back and we go look for you and we find you in the trunk of your car, no judgment. I don't know what you do in your spare time. But we find you in the trunk of your car, given the temperatures, you're going to be roughly car-shaped for a little while, right? And trunk-shaped. It's going to be a while before we can pull you out of the trunk and put you back in your seat like it's Weekend at Bernie's. Incidentally, you don't know what I'm talking about. Going up on your 80s movies, you should have a watch list for this class. Weekend at Bernie's, required material. You're welcome. So, why? What's that? What's it called? That is, of course, as if you've ever watched any sort of CSI-based forensic drama crap on TV, that's rigor mortis. The muscles lock when you're dead, at least for a while. Why? Well, because when decomposition sets in, and it does set in fairly quickly, the first thing that's going to sort of start decomposing is here at the Sarcoplasmic Reticulum. And then that calcium is going to leak out. And that calcium is going to bind the troponin. Troponin is going to move tropomyosin, and tropomyosin is going to expose those active sites. Myosin binds to actin, forms the cross bridge. Now what? There's no ATP. ATP breaks the cross bridge, but you're super dead. And when you're dead, you're not making any more ATP. So without that ATP, there's nothing to break the cross bridge. So it forms and it stays there until decomposition damages the fiber enough that you can move it again. And if they have evidence in their hand, you're going to have to break their stupid hand to get the evidence out of their hand. You might as well just cut it off. It'll slow down the fingerprint process anyway. The more you know. You're welcome. That's rigor, and that's because we're losing this. So ATP breaks the cross bridge. Without ATP, the muscle locks, and you get rigor. Fun with muscles. Like I said, this whole mechanism is essential. Acetylcholinesterase, we can't screw this up. And if we do, the results are disastrous. You can't breathe. More on neurotoxins and how they affect the neuromuscular junction later. In terms of what I expect you to know exam-wise, by no means Am I going to ask you to walk through this process step by step? That's not important. It's not important that you know step by step what happens in the cross-bridge cycle. Not important. What is important is that you can apply what you know about the cross-bridge cycle to something clinical, because that's what's going to come into play. Not that you know that, right, well, that you know that the order of ADP and or that inorganic phosphate leaving the myosin head, I've combined them in one step. It doesn't really happen like that, but it doesn't really matter. Big picture is our concern here. So that if I tell you I've blocked acetylcholine, you can tell me that's going to prevent contraction. Let's look at another example of that. We have a not uncommon disease called myasthenia gravis. Myasthenia gravis is an autoimmune disease. And the immune system then decides to attack those stupid acetylcholine receptors. And if you decrease their number of acetylcholine receptors, you get that. Anything that prevents acetylcholine from binding to a receptor, even if it's eliminating the receptor, prevents contraction. Keep in mind that your muscles are never fully relaxed. We talked about this with joints. We've always got muscle tone. That state of constant partial contraction. Well, if I start eliminating receptors, the result of that is going to be decreased muscle tone and muscle weakness. Sagging of facial muscles, because there's no tone there. Difficulty with manipulating objects. Now we can treat the symptoms. We can't actually cure this, like most autoimmune things, but we can treat the symptoms. How would we do that? Well, we can't put the receptor back, but we can add more acetylcholine to the synapse. So treatment here, adding more acetylcholine to the synapse, would be an acetylcholinesterase inhibitor. Now, we're not talking like organophosphate, but a mild acetylcholinesterase inhibitor adds more acetylcholine to the synapse. And any acetylcholinesterase inhibitor is going to increase tone. It's going to make that muscle contract more. This is not an uncommon disease. If I remember correctly, former First Lady Laura Bush has myasthenia gravis. If you watch daytime television on Days of Our Lives. Okay, so before I go any further with this, I need to justify this. When my youngest son was born, he was in the NICU, Texas Children's in Houston, and then he had to have surgery. And so you're in the waiting room and you're in the hospital for weeks and weeks. And daytime television is all that you've got. There's not much on. But if you have to watch daytime television and the soap operas come on, I fully recommend watching Days of Our Lives, because it's by far the stupidest plot line ever. Really, it needs to be a comic book or something. Even comic book plot lines are more feasible. But during this time period, one of the actresses, they wrote myasthenia gravis was part of the plot. Like this, the character had this disease and she was suffering from the symptoms of it and trying to go about her daily life as a whatever she was in the soap opera. And the first time I saw it, I told my wife, I'm like, that is a freaking good actress, because she is mimicking those symptoms just perfectly. I'm like, I don't even know who does her makeup. No, she actually has it. The actress actually has the disease, and she was symptomatic, and she came into work, and rather than just saying, hey, take the day off, they're like, you know what? Let's just write it in. Not like they got any more feasible plots than that. Let's do it. So yeah, she still had to work. That kind of sucked. So yeah, we'll look at, if I have time, we'll look at pictures later. But anyway, that's another issue there. So this is really what I'm looking for. I'm looking for you understanding that, yeah, and understanding the big picture here, not necessarily going through the step-by-step motion. I'll throw some animations of this up on Blackboard so that you can see those animations. I believe I've got, if I can, I'll add links to them so that you can just watch those animations of the process that's happening. But let's zoom out a little bit. So we've got this movement of this sarcomere. There's not much going there. Like, it's a minuscule movement of microscopic filaments past one another. And if we send just a single action potential through it, right, it's going to move and then stop. A single cycle of contraction, relaxation is called a twitch. Let's graph that. I can do better than that. Right? Much better. Contraction and relaxation. That way you have fast twitch fibers and you have slow twitch fibers. But you're not creating a lot of force there. No, a lot of movement there, right? It contracts and relaxes. How do we make the muscle fiber shorter then? Right, that's not full shortness, like that got like a fraction shorter and then it released. How do we get it shorter? Well, let's look at what the action potential looks like behind that. Your action potential then would be here. I can do much better than that. So that still sucks. So there's your action potential. There's your muscle twitch. Well, let's see what happens. Let's fire those action potentials. Let's send 2 of them through there. Fire 2 of them, what happens? The muscle fiber begins to contract. And before it can relax, you fire the next action potential. It relaxes. Now we got the fiber a little shorter. Let's put more of them there. Then what we get is this. It stair steps up. The faster you fire those action potentials along the cell membrane, the more of the stair stepping you get. It builds on itself. Before it can go through that complete muscle twitch, you're already contracting the fiber again, and that will happen up to this maximal point where it can't get any shorter. This is called temporal summation. Temporal summation. Temporal summation is increasing the frequency of the stimulus, and this causes increased tension in the fiber. If we were doing this in the lab, in person, what we would do is we'd take a frog and we would do something called pithing the frog. No, I don't have a lisp. what that means is that we take a needle and we push it through the foramen magnum of the skull into the brain. I'm actually demonstrating this in my hands, even though you can't see me. So I push it through the foramen magnum. I'm still doing it. I can't stop. Into the skull, into the brain of the frog, and then we scramble it around, destroying the frog's brain. Then we pull it back and we push it through the vertebral foramen of the spine, and we do the same thing. We destroy the spinal cord. Now we've destroyed the frog's entire central nervous system. But as an amphibian, a lot of respiration happens through the skin, so we're oxygen's going back and forth through the skin, so the heart's going to keep beating, and the tissues remain alive, even though the frog is super dead because you destroyed its brain. Let me get this clear from this point forward. The frog is dead. This is just as easily achieved by decapitating the frog and then pushing the needle straight down and destroying the spinal cord. Moving on. Super dead, not torturing a living frog. Super dead for science. You're welcome. So now what we would do is, with the frog dead, we would open up its leg and expose the sciatic nerve, a big nerve going through the leg. And we would hook up electrodes to that nerve. And then we would hook those electrodes up to a muscle inducer, which is then hooked to this big battery. We would set it on 1, and you might have you push the button. And when you push that button, a single electrical impulse will travel along the sciatic nerve down into the muscle, and you will see the muscle twitch. It will contract and relax. Then I'll have you turn up the frequency so that when you hit the button, you don't just get one impulse, you get rapid fire impulses. And the faster you fire it, the shorter that fiber gets, the more tension you build in the fiber, until you get to this point where you've reached the maximum contraction. The fiber can't get any shorter. It's at the maximum contraction level. It's as short as it can get. Incidentally, where you have that and you get that maximum contraction of the muscle, it is not getting any shorter. That is tetany of the muscle, or tetanus, not to be confused with the disease. So tetany, sustained contraction. So we vary the length of the fiber. When we contract muscle, we vary the length of those muscle fibers by varying the frequency of the stimulus from the brain. But muscles aren't just one fiber, right? There's a bunch of fibers. And each of those fibers has a neuromuscular junction. Fibers are in things called motor units. A motor unit is a single neuron all the fibers it innervates or controls. So you'll have one neuron that branches off like 10 or 100 times or whatever. And then out here you've got your muscle fibers. So you have like one neuron that controls like 5 or 6 muscle fibers. Now, like I said, even under maximum effort, you never use more than like a third of them simultaneously. How do you decide how many of those fibers to use? Well, the more fibers you use, the more force you can produce. And this depends on the signal from the brain. This is called multiple motor unit summation, or recruitment. You use less muscle picking up an egg than you do picking up a car. Well, that makes sense. Why? It's controlled by your brain. When you pick up an egg, you don't smash it in your hand. Hulk smash egg, Hulk hate egg. Right? You can pick it up without doing that. But then you can go outside and try to lift your car. You see these things like where these people like lift cars off babies or whatever. And that's a thing. How does that work? Multiple motor unit summation. Most people can't pull this off, but sometimes under a life and death circumstance, your brain can just throw a Hail Mary pass and contract all the fibers of that muscle simultaneously and create a hell of a lot of force all of a sudden. That's multiple motor unit summation. Now, normal basis, it's not that drastic, but basically the gist of this is you use less muscle when you don't have to produce as much force. And the more force you try to produce, the more muscle fibers you recruit. Now, there is an order of recruitment. The order of recruitment, we use the smallest fibers first. So you start with small fibers. If that's not enough, you recruit some friends. If that's not enough, you recruit some friends. And you keep getting bigger and bigger fibers recruited in this until there's enough fibers in play to do the job you need them. That's multiple motor unit summation. Multiple motor unit summation, increased intensity of the signal the brain recruits more motor units. If we were doing this in lab, what we would do is we would turn up the voltage on that inducer. And the more voltage you apply, the more of that muscle you'll see fire. You'll see much more drastic contraction at a higher voltage because more of the fibers fire when you send more electricity through that nerve. It's funny because the frog will move drastically and hilariously for science. It's cool. Temporal summation, multiple motor unit summation. Now, there are different types of contraction in the muscle. We have isometric, isotonic contraction. Isometric, iso is same, metric is length. So this is a contraction without a change in length. Think about going out and trying to lift your car. You're not going to lift your car, but you can try. And you can feel the tension increase in the muscle. So we get increased tension, but no actual movement. That's isometric. Isotonic contraction, tonic is tension. So this is contraction. With constant tension. And there are two types of isotonic contractions. There's concentric and eccentric. Concentric, muscle fiber gets shorter. That one's easy to understand. Flex your biceps muscle. Put your hand on your biceps when you flex your arm. Constant tension. Eccentric, the muscle gets longer. Now relax that biceps muscle, go back to resting position. Even though the muscle fiber is getting longer, you can still feel constant tension in that fiber. It's a latching and letting go and latching and letting go and latching and letting go until you get back to the 0 point of your arm, until you get back to your arm being extended. That's an eccentric contraction. So concentric, the muscle fiber gets shorter. Eccentric, the muscle fiber gets longer. All right, so we're going to pause here and Then we're going to come back and talk about different fiber types of muscle, different types of exercise briefly, and then cardiac muscle and smooth muscle briefly as well. This is the bulk of muscle fiber stuff, and we'll touch on this a little later again and kind of come back to it and go over it.